Students, today we are going to see in Science-2, Chapter 3, Life Processes in Living Organisms, Part 2. Today we are going to see the Part 1 of this chapter. We have studied various life processes in previous classes. All those life processes like nutrition, respiration, excretion, sensation and response, etc. are essential to each living organisms to remain alive. Beside this life processes, one more process occurs in living organisms. It is reproduction. However, reproduction does not help the organisms to remain alive, but it helps to maintain the continuity of the species of that organisms. So, what is reproduction? Formation of new organisms of same species by earlier existing organism is called as reproduction. Reproduction is one of the various important characters of living organisms. It is also one of the various reasons responsible for evolution of each species. In living organisms, reproduction occurs mainly by two methods, and those two methods are asexual and sexual reproduction. What is asexual reproduction? That's process of formation of new organisms by an organism of same species without involvement of gametes is called as asexual reproduction. So in this reproduction it does not involve union of two different gametes. The new organism has exact genetic similarity with the reproducing organism. This is uniparental reproduction and it occurs by mitotic cell division. Absence of genetic recombination is the drawback, whereas fast process is advance of this reproductive method. Now we shall see the first asexual reproduction in unicellular organisms. Let's see binary fission in amoeba. Amoeba is the single cell eukaryotic organism with no definite shape and moves by means of pseudopodia, and it belongs to the kingdom Protozoa. Amoeba shows extremely diverse in nature, so they can produce using varieties of different methods such as binary fission, spores and even sexually. Binary fission is the most common method asexual reproduction employed by amoeba. During this stage, amoeba stops its movement by withdrawing its pseudopodia and forms spherical shape. The genetic material is divided by the process of mitosis. After the division of cytoplasm follows the division of the nucleus, resulting in two daughter cells which are identical clones obtained by parent cells. Next, we shall see transverse binary fission in paramecium. Now, as we know, children, binary fission occurs in transverse fashion in paramecium. It is a common method of reproduction in paramecium which occurs during favorable condition. A fully grown paramecium is divided into two daughter individuals. Division occurs at the right angle to the longitudinal axis of the body. Nuclear division is followed by the cytoplasmic division. Paramecium stops feeding before initiating binary fission and then its oral groove disappears. Micronucleus divides by amitosis and micronucleus divides by mitosis. After division, they move towards the opposite end. At the same time, the constriction develops at the middle part of the body, which divides cytoplasm into two equal parts. In this way, two daughter paramecia are produced. Now we shall see longitudinal binary fission in EUGLENA. Binary fission is the most common method of asexual reproduction in protozoans. Longitudinal binary fission is performed by EUGLENA. In this type of binary fission, the body divides into two halves longitudinally, hence called longitudinal binary fission. During this process of binary fission, the nucleus, basal granules, chromatophores, cytoplasm undergo division. The nucleus divides by mitosis into two daughter nuclei. Then kinetosomes and the chromatophores they also divide. At first, a longitudinal groove develops in the middle of the anterior end. This groove extends gradually towards the posterior end until two daughter individuals are separated. One daughter euglena retains the parental flagella, the other daughter individual develops new flagella from the newly formed basal granules. The stigma, paraflagellar body, and the contractile vacuole of the parent disappear. They develop afresh in both the daughter euglenae. The longitudinal binary fission is known as symmetrogenic division because the two daughter euglenae resemble each other like mirror images. Next, we shall see multiple fission in amoeba. As earlier, we had seen binary fission in amoeba. Now we shall see multiple fission in amoeba. Asexual reproduction by multiple fission is performed by amoeba and other similar protists. Amoeba stops the formation of pseudopodia and thereby movements. Whenever there is lack of food or any other type of adverse condition, it becomes rounded and forms protective covering around plasma membrane. Such encysted amoeba or any other protist is called as cyst. Many nuclei are formed by repeated nuclear divisions in the cyst. It is followed by cytoplasmic division and thus many amoebae are formed. They remain encysted till there are adverse conditions. Cyst breaks open on arrival of favorable conditions and many amoebae are released. Now we shall see budding in yeast. Bring the active dry yeast powder from market. You take 50 ml of warm water in a conical flask and just add 5 gram of active dry yeast powder and 10 gram table sugar so that water and mix with the mixture. Keep the flask in warm place and after an hour, just take a drop of that mixture on a clean glass slide. Put a cover glass on that drop and observe it under the compound microscope. You will see that the cells performing budding, that is, a small bud coming out of many parent cells. As you can see, asexual reproduction occurs by budding in yeast, a unicellular fungus. Each cell reproduces two daughter nuclei by mitotic division so as to reproduce by budding. This yeast cell is called as parent cell. A small bulge appears on the surface of the parent cell and this bulge is actually a bud. One of the two daughter nuclei enters this bud after sufficient growth. Bud separates from the parent cell and starts to live independently as a daughter yeast cell.